to NPTEL coursework. This is an extra session. So this is the re-recording for the video upload. This is an extra session for the course Experimental Biotechnology. It is provided by Professor Vishal Trivedi from IIT Guwahati, and I'm one of your TA, Sumati from University of Hyderabad. So this session I have chosen to discuss the topics from the assignment of week 0. So let's go into those. So the first topic regarding this was chromogenic HRP based substrates. So the chromogenic HRP based substrates, they react with HRP, that is horseradish peroxidase. It is an enzyme, and in the presence of H2O2 it produces a colored insoluble or soluble product. So basically the color helps us visualize or quantify the concentration. So these colored products can be visualized directly by naked eye or under a microscope. So certain examples like 4-chloronaphthol, 4CN, it is a chromogenic substrate, and reaction that takes place is HRP catalyzes oxidation of 4CN in the presence of H2O2. This results in a blue to purple precipitate, and it is widely used in methods like Western blot, immunohistochemistry, etc. So, where the colored precipitate then localizes the HRP enzyme and therefore the antigen, so we can quantify the presence or quantity of the antigen present based on the color intensity or the presence of the color. Other than this, there is also DAB. DAB is also widely used in immunohistochemistry. It is 3,3 diaminobenzidine, and this is also a chromogenic substance. So the reaction is when HRP converts DAB into a brown insoluble polymer, uh, which occurs in the presence of H2O2. We can see the color based on the presence of the antigen or the amount of the antigen. So this results in a brown precipitate forms at the site of enzyme activity. Then it's widely used in immunohistochemistry for permanent tissue staining and light microscopy. So once we stain the tissue sections, we can visualize it using a light microscopy in the RGB category, which will help us visualize the structural details and as well as the amount of antigen that we are looking for. The brown stain marks the presence of the target protein. So this is 4CN and DAB. Other than this, there is TMB, which is 3355 tetramethylbenzidine. So this is also similar to DAB, and it is also chromogenic substance. The HRP catalyzes the oxidation of TMB with H2O2. Solution turns blue here, which changes to yellow when the reaction is stopped with an acid. So this is widely used in assays like ELISA, uh, which is a quantitative detection using a spectrophotometer. The absorbance that is used is usually taken at 450 nanometers, and this is very commonly used in ELISA. Other than this, we have luminol. This is not chromogenic. So here it is a chemiluminescent reaction. So the chemiluminescent substrate and not chromogenic substrate. But here also we have HRP. HRP catalyzes oxidation of luminol by H2O2, resulting in light emission. So this is captured or detected at 425 nanometers. Here, instead of producing a visible color change, the reaction produces light, that is luminescence photons, which are then detected using a detector, and the image is captured. So it is commonly used in chemiluminescent Western blotting, where emitted light is detected by X-ray film or a CCD camera. It is highly sensitive and it allows the detection of even very low level of target proteins. So this was about luminol. So out of the three options, only luminol is not chromogenic, uh, HRP. The remaining all are chromogenic. So this is a comprehensive table of all the three options. So these three, 4CN, DAB and TMB are all chromogenic and produce a visible color which can be detected either by microscopy or spectrophotometry. Then luminol is chemiluminescence, so there is light emission which is detected by a film or a camera. Here there is no color that is produced visibly. Next is regarding SDS page. So SDS is sodium dodecyl sulfate and page is polyacrylamide gel electrophoresis. So it is widely used to separate proteins in a mixture and resolve proteins of different molecular weights. The separation principle is based primarily on the molecular size of it. And let's see the step-by-step methodology. So first is denaturation by SDS. So SDS is used to denature and also it prevents the folding of the protein into its 3D structure. So proteins are treated with SDS. It is an anionic detergent which binds along the polypeptide chain. So approximately 1 SDS per 2 amino acids is bound, and SDS unfolds the protein from its native form and destroys their native 3D structure. So this helps the proteins travel in their simpler form into the gel. Then there is uniform negative charge that is also imparted. So SDS coats proteins with a uniform negative charge density. This masks their native charge. So all of them have similar charge, and the separation is only based on the difference in their molecular size. So as a result, the charge to mass ratio becomes nearly constant for all the proteins. Then it also helps in linearization. So heating is done, and often beta mercaptoethanol or DTT is used, which breaks the disulfide bonds, further ensuring the proteins are linearized. Then separation is done, like we said, based on the size. So during electrophoresis, proteins migrate through the polyacrylamide gel matrix. Since charge differences and shape are removed here, that is neutralized, the rate of migration depends solely on the size of, and the smaller proteins move faster and travel farther than the larger ones. So we get a gradient of uh, high molecular size proteins to low molecular size in a descending order. And what is native page? So this was SDS page, which is a denaturing environment. And native page, we do not use denaturing and agents like SDS. So it is just a polyacrylamide gel electrophoresis under native conditions. It is a technique in which the proteins are separated without denaturing agents like SDS or urea. So as a result, the proteins retain their native 3D conformation, and they are also biologically active. Their natural charge and shape is retained. The ability to interact with other biomolecules is also retained. So this can be used in different methods. So this makes native page very useful for studying functional and structural properties of the proteins in their biological form. Principle of separation of native page. So unlike SDS page, which separates the proteins only by the size, native page separates proteins based on a combination of their size, shape, and net charge. So size is the molecular weight, shape is its 3D structure or its native form, and the net charge is again dependent on the pH because of the pI and composition. Because these properties influence the migration speed in the gel, proteins with different native states migrate differently. So native page can be used to study the different characteristics of a protein, either its oligomeric nature, protein-protein interactions, gel catalytic activity, and 3D structure of the protein. So to study protein-protein interactions, because the proteins stay in their native conformation, they can form complexes with other proteins or ligands. So when the protein is its in native conformation, it can bind with its interacting partners. If you denature it, it will not bind. So SDS cannot be used for this purpose. Only native page can be used. So these complexes often migrate differently, slower than the individual monomers of the same protein. So once denatured, the protein splits into its individual monomers. Generally, proteins exist as oligomers or heteromers in their native form. So this will disintegrate that in SDS page, but in native page you do not see the disintegration. The protein, if it's a tetramer, it will be a tetramer. If it's a pentamer, it will stay a pentamer. So they will obviously migrate slower. So for interaction studies, we can also detect transcription factor DNA or antibody antigen complexes. And we can also, like I mentioned, we can study the oligomeric states, whether a protein exists in its monomeric form or its dimeric form or a multimeric form. Then we can also monitor in-gel catalytic activity of the enzymes. So since proteins retain their enzymatic function, you can perform activity staining directly in the gel. So this is zymography or dehydrogenase assay. So in zymography, detecting protease activity in the gel after electrophoresis can be done. In dehydrogenase assay, detecting oxidoreductase activity with colorimetric substrates can be done. So other than this, we can also use it to determine the 3D structure of proteins. So native page does not give atomic level 3D structures, like you cannot like visualize the structure, or we cannot like get minute details of the structure like crystallography or cryo-EM, but we can get an information about the quaternary structure, whether it's a monomer, multimer, or the oligomeric size of it, the conformational changes that it can undergo. So, for example, if there is a shift in the migration, it suggests that there is conformational change or a complex formation. So, you may not be able to detect what type of change it is, but you can understand that uh, there is a change, or there is a complex formation, or it has the ability for complex formation. Next is regarding the SDS page loading die. So there is a specific die that we use for loading the samples. So before proteins are located into the SDS page gel, so before the proteins are loaded into the SDS page gel, they are mixed with a loading dye, which is also called as a sample buffer or lamellae buffer. So this buffer is carefully designed mixture that prepares the protein sample and gets it ready for the electrophoresis. So what it does is it denatures the proteins, so they migrate based on only the size. It provides uniform charge, so charge based bias is also not there, and it allows easy visualization of sample loading. So the sample protein or anything will generally be translucent. You cannot properly visualize it when you load it into the gel in the buffer. So this also has dye in it, which helps us visualize our sample and load it properly into the gel. And also it helps us visualize the sample while it's running, or while the migration is taking place in the gel throughout the electrophoresis. So this ensures that the sample stays at the bottom of the well, because it also has a density providing material like glycerol. So this helps the sample to gain more density and settle into the well. Otherwise, if you do not have a dense material with it, it will diffuse into the buffer and there will be loss of sample. So these are all the important characteristics of a loading die. Then the main components of loading die are tris HCl, which has a pH about 6.8, and SDS and beta mercaptoethanol, bromophenol blue, and glycerol. So tris HCl is the buffer which helps maintain the pH in the sample and in the buffer environment, and SDS is for denaturing the proteins, and it imparts a uniform negative charge. Beta mercaptoethanol or DDT reduces the disulfide bonds between the proteins. And then bromophenol blue is the dye, it's the tracking dye which helps us monitor the sample throughout the migration. Then glycerol is the one that increases the density, so it increases the sample density to help aid the loading of the sample and also help the sample to settle nicely in the well. So, what is exactly the role of glycerol and why it is added? This helps in increasing the density because it itself is a high density material. It is highly viscous and it is a polyol. It's a sugar alcohol. So its inclusion in the dye serves more to impart a physical property rather than any kind of chemical effect. Then it increases the sample density. Electrophoresis is done in water that is in the buffer. So the density is close to 1 gram per ml. Glycerol has a much higher density, which is 1.26 gram per ml. So when you mix glycerol into the loading buffer, the final sample becomes denser than the running buffer. That is the major idea behind it. And this density difference ensures that the sample sinks straight to the bottom of the well rather than floating or diffusing away during loading. So it prevents sample loss. And without glycerol, the sample might get dispersed into the buffer. So as soon as you pipette it into the well, causing uneven loading or poor band resolution. So it also, so therefore it will help in accurate pipetting and loading. When you want equal amount of proteins into the gel, this is very important. So glycerol makes the sample viscous and helps it stay as compact as a droplet. And this makes the process of pipetting or sample loading easier and more precise, especially for smaller volumes. Then eventually it will help in preventing the sample loss, because when the sample sinks, it stays in the well and more protein enters the gel, improving the detection sensitivity. This is particularly important when we load very low concentration of proteins. So when preparing SDS gel, we have two types of gels in it. One is the resolving gel and one is the stacking gel. So we will talk about that here. And the resolving gel is the one which is added first. So it is the lower portion where you actually separate your protein of interest based on the size. So when preparing an SDS gel, the resolving gel is poured first. During its polymerization, it is a liquid mixture which contains acrylamide and bisacrylamide, which is already usually prepared. 30% acrylamide is added. Monomers, so these have the monomers that form the polyacrylamide matrix. Then we have buffer, tris buffer, which helps maintain the pH. We have SDS, which ensures denaturation and uniform charge. Then we have APS, which is an initiator. Then we have TEMID, which is a catalyst for free radical formation. So APS helps in the polymerization process, and when TEMID is added, free radicals are released, and this helps increase the reaction at a faster rate. So the problem here is oxygen inhibits the polymerization. So the polymerization of acrylamide is a free radical reaction. So oxygen from the air reacts with the free radicals, quenching the reaction and inhibiting polymerization, especially at the top surface of the gel exposed to air. So as a result, the gel may not solidify properly or form an uneven layer at the top. So what we do for this is we overlay an organic solvent to prevent oxygen from diffusing into the gel. So if it diffuses while polymerization, we will get an uneven layer. So a thin layer of immiscible organic solvent such as N-butanol or isopropanol is gently added on the top of the resolving gel. So this seals off the surface of the gel and thereby preventing oxygen entry. So also we do not need any bubbles during the preparation of the resolving gel. So in case while pipetting it out, if there are any bubbles at the top, adding this immiscible liquid will also help eliminate that buffer and eliminate the air present in it. So after polymerization is completed, the solvent is poured off, that is the butanol or isopropanol that is added is removed from the gel, and then the stacking gel is casted on top of the resolving gel. So this is how we can avoid the oxygen inhibition of polymerization. So next is a native page. It is under the non-denaturing conditions. So like we discussed, the proteins retain their native structure, 3D folding, quaternary structure, and enzymatic activity are preserved. So no SDS or denaturants are used in this case. So the principle of migration in native page is that the proteins migrate in an electric field due to their net charge. The rate of migration depends on three things mainly. First is the net charge of the protein in the at the running pH, size of the protein, shape and conformation of the protein. So essentially, proteins are separated based on their charge to mass ratio here, whereas in case of SDS page, the charge to mass ratio is usually constant because of the addition of denaturants, which will neutralize the charge based bias. Then, in case of native page, highly charged small proteins move faster, large or less charged proteins migrate more slowly. Proteins with the same molecular weight but different charges can be separated. In SDS page, this is not possible because the charges will be constant and it's only molecular weight. So if the proteins are of the same exact molecular weight, you cannot resolve it into a different area on the membrane. But in case of the native page, because they have charge difference as well, their movement or their migration will be slightly varying. So the proteins with same charge but different sizes will also migrate differently. So the proteins with same molecular weight and different charge will also be separated, and vice versa, that is the same charge and different molecular sizes will also be separated. So this is like a brief of SDS and native. So SDS is denaturing, native is non-denaturing. In SDS, the separation is based only on the molecular weight. In native page, it is based on the charge to mass ratio, size, shape, and the conformation of the proteins. So next is a problem. So where protein X has a molecular weight of 80 kilodalton and it is a heterodimer. So how many bands will you observe on a gel if you resolve the protein on SDS and native page separately? So it's a heterodimer, that means it has two different monomers, and it is 80 kilodalton. So in SD's page, what happens is the protein gets denatured and it will lose its heterodimer form, that is the quaternary structure. So the subunit associations are disrupted. So each subunit will run according to its individual molecular weight. So what it results is, if the heterodimer has two different subunits, say for example a 50 kilodalton subunit and a 30 kilodalton subunit, two individual bands will appear. And in case of native page, since it does not denature the protein or the quaternary structure, you will see only one single band at 80 kilodaltons. So in case of SDS page, you will observe two bands if it is because it is a heterodimer, and if it is in native page, you will observe a single band. So here the answer would be SDS and native. So it is two bands and one band. Next, yeah. So the major key points to remember for native page, it does not affect the uh, conformation of the protein. It doesn't denature the protein. Proteins retain their native structure, including the subunit association, even though they are heterodimers or homodimers or oligomers. Then migration depends on the charge, size, shape of the intact protein complex. Implication for the heterodimer is it remains intact in the native page, and you will see it as a whole protein complex when run on a page gel. Next is the use of a tracking dye. So we saw that we use tracking dye in the sample preparation, and it is added to give color and for visualization. So a tracking dye is a small colored molecule basically, and it is added in the sample buffer before loading the protein into the gel. So its main purposes are to monitor the electrophoresis progress, that is to visualize when your sample is migrating through the gel, and help the visualization during loading, and acts as a front marker. So for monitoring the electrophoresis progress, the dye migrates through the gel ahead of the proteins. So basically, since it is a small molecule, it will run forward ahead of all the most of the proteins present in your sample. Then when the dye is nearing to the bottom of the gel, you know that your run is completed. So it helps you know when to stop your electrophoresis process, so that all your proteins have been resolved. And also it will help not lose your lower molecular proteins, because of its smaller size, that is the one which will travel faster. And usually we wait till the dye is released from the gel, so that we can have all the proteins in the gel. So this makes it easier to see the sample also while loading. So when you load it, it will sit nicely into the well because of the glycerol that is present. But also you can visualize whether your sample is being diffused out from the well, or it has sat into the well properly like a droplet. Then it acts as a front marker, that is a dye front, that ensures that no proteins run out of your gel and you don't lose your smaller molecules. So we also used bromophenol blue as a dye in the tracking dye. So it is a very small molecule which is about 670 daltons, and it is blue in color and it has high visibility. And the charge of it is negatively charged under sample buffer conditions, so it migrates towards the anode. So it will run along with your protein sample but in the front. So the migration is it moves faster than most of the proteins in your SDS page. So anything about, since it is 670, so anything about that, like your even 5 kilodalton, your mostly proteins are in your kilodalton, and your 5 kd, 1 kd, all these will be just above your diaphragm. So your migration of your dye is always faster than your protein, so it will always be ahead of your actual protein bands. So what are the advantages of it? It is inexpensive, it is easy to visualize, it is easy to detect with naked eye, and it is chemically stable and therefore is a standard in SDS page conditions. And it also does not interfere with your proteins, and it does not interfere with the separation, which is very important. And essentially the bromophenol blue is a visual guide, and it's not a reagent, because it does not affect your protein mobility or staining or anything. Then this is a comprehensive table about it. So other dyes which are not suitable. So bromophenol is more suitable in the given options here. Other dyes which are given are not suitable because they are fluorescent. Like FITC is fluorescein isothiocyanate, and it is covalent. It covalently binds to the proteins, and it cannot be visualized with naked eye, and you have to detect it. So this is not used in gel, first of all, because it binds to your proteins. Secondly, the next option that was given was Comasi Brilliant Blue. So this also stains the proteins. So it is used after the electrophoresis for visualization of your protein bands and not while running your gel. So it does not migrate through the gel, and it is not visible until staining. Then the other option that was given was rhodamine dextrin. So this is also a fluorescent marker. It's usually for high molecular weight standards or gels, and not commonly used for routine SDS page. These were the other options. So the correct option for this is bromophenol blue. So yeah, so that was about the use of bromophenol blue. So how it works in practice is bromophenol blue is added to your sample in the lamellae buffer, and we mix the protein samples throughout with SDS, beta mercaptoethanol, DDT, glycerol, tris buffer, etc., etc. And excuse me. So then after that we load it into the gel. And during electrophoresis, the bromophenol blue moves ahead of the proteins. It helps track the progress without removing the gel from the apparatus. And we stop the run when the dye is near to the gel, or when it is released from the gel. Proteins will still be within the gel, and the small size of it ensures that it does not co-migrate with the proteins, preventing overlap with the bands. So therefore, it is a standard tracking dye in SDS page. It's a visual indicator in electrophoresis progress, sample loading, and this is how it is very useful in SDS page. So next is SDS page. Uh, it separates the proteins primarily based on the molecular weight. So SDS, what is the role of SDS in here? So SDS, like we discussed, is a denaturing agent. It's an anionic detergent. So it binds along with the polypeptide chain. Approximately one SDS molecule binds to two amino acids. So this also linearizes the protein, disrupting the secondary, tertiary, and quaternary structures. Then imparting uniform negative charge, SDS coats the protein with negative charges regardless of the protein's original charge. So basically all your proteins will be negatively charged. This results in that all the proteins have approximately same charge to mass ratio. Because charge is now proportional to the mass, proteins will migrate only according to their size rather than their native charge. So how it works in the gel is as we have the electrode set up with the cathode and the anode, the negatively charged SDS protein complexes migrate towards the anode. Larger protein molecules migrate slower through the gel matrix, and smaller proteins migrate faster. So basically, it is a denaturing agent. It impacts negative charge uniformly, and it helps in maintaining the linear form of the proteins. Next is urea page. So urea page is a form of polyacrylamide gel electrophoresis in which the gel and the running buffer contains a high concentration of urea, typically around 6 to 8 molar. Urea So urea is a chaotropic agent, meaning it disrupts hydrogen bonds and non-covalent interactions in the proteins. So without breaking the covalent bonds like disulfides, unless reducing agents are added. So unlike SDS page, urea does not fully denature the proteins or impart uniform charge. It only partially unfolds the proteins. So it leaves some of the structural elements intact. So what is the principle behind it is why uh, urea page can study folding states. Partial unfolding of proteins takes place. So proteins have secondary structures like alpha helix, beta sheet, etc., and tertiary structures stabilized by the hydrogen bonds and hydrophobic interactions. So urea disrupts the non-covalent bonds, causing gradual unfolding of it. Then electrophoretic mobility reflects the folding state. So folded proteins are more compact. They migrate differently than the unfolded or partially folded proteins. Partially unfolded proteins have altered charge distribution and hydrodynamic volume. This affects their migration in the gel. So a controlled denaturation is done by varying the urea concentration. So we can observe the unfolding transitions. So this is useful for studying the folding kinetics, stability, and also the intermediate states of the transitions. So the application, the major applications of urea page, uh, protein folding or unfolding studies can be done. We can determine how the proteins respond to the denaturants. We can compare the stability of mutant versus the wild type proteins. Then detection of conformational heterogeneity can be done. So different folded states or isoforms may migrate differently. So this can be used to study its conformational change, and nucleic acid studies can be done. That is, we can also use it to denature RNA or single-stranded DNA to study secondary structures. So how it works in practice is we can prepare the gel containing 6 to 8 molar urea. Then we have to prepare the samples, native or partially denatured. Then load into the wells and run under non-denaturing conditions or mild denaturing conditions. So then we observe the migration patterns, first fully folded, then compact, which are compact, migrate faster. Partially unfolded gets extended, so they migrate slower. Then we can also titrate the urea concentration to determine the denaturation midpoint, which is CM, which is an indicator of protein stability. So the major key takeaways here are in urea page, partial denaturation is done to study the folding and unfolding states of the proteins, provides information about protein stability, conformational changes, not primary sequence, but the 3D atomic structure. Uh, so the atomic level details cannot be detected, but we can just know the difference in their conformational change, whether it is there or not. So the migration depends on size, shape, and partial unfolding, making it a very powerful tool for protein folding studies. Next is regarding PVDF. So PVDF is used popularly in Western blotting. It is polyvinylidene fluoride and is very commonly used membrane during Western blot to transfer the proteins from your gel. So it has a high protein binding capacity, especially for hydrophobic proteins. It is mechanically strong and it can withstand multiple washing and stripping. So stripping is done when you want to reprobe your blot or your membrane. So once you add a primary antibody for your protein of interest and you have visualized it, if you don't have your sample or if you want to probe another protein in the same membrane, you can do a process called stripping, which will remove the antibodies bound to your proteins, which will help you, add a different antibody for a different target protein in the same membrane. So PVDF is very useful for that. and it is compatible with various detection methods like chemiluminescence, fluorescence, and also calorimetric. But the main challenge is PVDF is intrinsically hydrophobic itself. So dry PVDF membranes repel water and aqueous protein solutions. So protein will not bind efficiently unless the membrane is completely activated. So we use ethanol to activate or pre-wet the PVDF membrane before the transfer is done. So methanol is used to activate it, and how it works is complete immersion has to be done in 100% methanol, and the membrane becomes translucent after it gets completely soaked in methanol, indicating that it has been properly wetted. Then methanol interacts with the hydrophobic PVDF, allowing it for transfer of aqueous or polar proteins and effectively penetrate into the membrane. So then we rinse it with water or the transfer buffer, which removes the excess methanol and equilibrates the membrane with a conductive aqueous environment for electrophoretic transfer. So as a result, the PVDF surface becomes hydrophilic enough for the proteins to bind on it during electrotransfer. Methanol also helps the proteins interact with the hydrophobic binding sites on the PVDF membrane and enhances the transfer efficiency. So why methanol is critical? Because without the methanol, proteins pass through the membrane or bind unevenly, and the transfer will not be efficient. And Western blot, when you develop it, the blot signal may be weak, patchy, or inconsistent. So methanol also helps stabilize the proteins on the membrane after the transfer, which reduces the blurring effect or smearing of the bands, so that when you detect it, you can get a nice crisp, clean band. So the solvent that is used for activating it is basically methanol. Water cannot be used because it cannot wet the hydrophobic PVDF alone, and protein binding will still remain poor. DMSO is not used because it may dissolve the hydrophobic polymers and damage your PVDF. And DMF is also not used because it's toxic and it is not required, and it is not used anywhere here in Western blot. So the actual uh, chemical or actual liquid solvent that is used is methanol. So practical protocol is to cut the PVDF membrane according to the size of your gel, immerse it briefly into 100% methanol until it turns translucent. It can take anywhere between a couple of minutes to several minutes, and rinse with water or transfer blot, and then place in the transfer apparatus for SDS page protein transfer. And after this step, the membrane is ready to receive proteins during electroblotting. So basically, to summarize it, PVDF is hydrophobic, so proteins cannot bind efficiently when it's dry. So methanol is used as a pre-wetting agent, which makes the membrane hydrophilic and ensures efficient protein binding. So this step is essential for high quality and high reproducibility of the Western blots. Next is regarding the biosafety levels. So I wanted to discuss this as well in this. So biosafety levels are between 1 to 4, that's 1, 2, 3, 4, and every level has its own specificities. So depending on it, you can classify certain agents or certain levels as a different biosafety level. So, biosafety levels are a series of protective measures established to prevent laboratory acquired infections and contain infectious agents according to their risk. They are standardized by organizations like CDC, USA, or WHO. So we have four main biosafety levels: BSL-1, BSL-2, BSL-3, and BSL-4. So a BSL-1 level is basically for agents not known to cause disease in healthy adults. Minimal risk to laboratory personnels and or the environment of the lab. So the key features are the agents that are used here are non-pathogenic, like E. coli, K12, Bacillus subtilis, non-pathogenic yeast, etc. So the lab practices here involve standard microbiological practices. No special containment is required. Basic hand washing and no edibles, no eating, drinking in the lab, and proper basic sterilized conditions. Then safety equipment required for this is minimal. Open bench work is also allowed. Gloves are optional but recommended. And the facility design is in such a way that a standard lab bench is there and sink, and no special ventilation is required whatsoever. So it is very minimal, but still we have to maintain basic microbiological sterilization practices. So minimal risk, standard lab hygiene is sufficient, and the goal is to teach basic microbiology safety. Next is the BSL 2 level. So this is an intermediate level, and this is for agents. So this is for agents with which are associated with human diseases, but usually mild and may possess a moderate hazard. So basically, the agents here are, it might be hazardous at certain conditions or when infected, but they are not completely toxic, or they are not lethal. So agents that can be used here are Staphylococcus aureus, Salmonella species, hepatitis B virus, HIV, which is inactivated, can also be used in BSL 2, but it has to be completely inactivated and made sure of it. So the lab practices here are BSL 1 plus restricted lab access, biohazard warning signs, proper disposal methods of contaminated materials. And the safety equipments required are gloves, lab coat, eye protection, biological safety cabinets for aerosols, etc. And facility design is, you need an autoclave here for sure. So autoclave availability must be checked, and sinks and hand washing must be done. Then self-closing doors are recommended. So here the risk is moderate, so the personnel can handle pathogens with proper training. And BSL 2 labs are the standard for many clinical and teaching labs. So here we have uh, agents that are associated with human diseases, usually mild but may possess a moderate hazard. Next level is BSL 3. This is a higher level. So here it is the agents which are used can cause serious illness or potentially could be also lethal if infected, even via inhalation. So it has a risk to the personnel and the environment is very high. So here, most when you use pathogenic microbes like Mycobacterium tuberculosis, SARS-CoV, Bacillus anthracis, etc., all these can be very serious and can cause lethal diseases. So they are under high safety level management. And the lab practices done here are BSL 2 practices plus controlled access, decontamination of all the wastes, specialized training in handling, and pathogenic aerosols must be handled by only trained personnel. Then safety equipment is class 2 or class 3 biological safety cabinets, respirators, and sealed containers. The facility design involves negative air pressure rooms, that is, air flows into the lab and not out of the lab. HEPA filtration is a must. Self closing doors are required. Separate laboratory air supply and exhaust is also required. So, it is, since it is high risk airborne pathogens are also used here. The strict engineering controls should be done, and PPE kits are mandatory. And the personal training is also is highly essential because it prevents exposure to it. So this is about the high risk BSL 3. Next is BSL 4, which is maximum containment. This is the highest level of life threatening disease causing agents. So, for dangerous exotic agents with high risk of life threatening disease, often which do not have a treatment or a vaccine yet, these are done at BSL 4, and maximum biocontainment is required. So the agents used here are like Ebola virus, Marburg virus, Lassa virus, etc. So the lab practices that are carried out here are BSL-3 practices plus a full body air supplied positive pressure suits, so that there is no exposure to the environment into the lab, and complete decontamination of all the materials and personals upon exit. Safety equipment are class 3 biosafety units, cabinets, fully sealed suits, chemical shower decontamination must be done. Facility design is isolated building, dedicated air supply and exhaust is very important, and HEPA filtration is mandatory. Secure entry and secure exit must be there, and a full complete waste decontamination might be done. So the pathogens used here can cause epidemics if they are not properly decontaminated. So they can be lethal to the personals and lethal to the environment and the surrounding population here. So BSL labs are highest risk pathogens. They we will use the viruses or the pathogens which do not have a treatment or a vaccine yet. So personals must be highly trained. BSL-4 labs are very rare and they are highly secure and are designed to prevent any sort of accidental release from the facility design. So this was about it, and this is a comparison between BSL 1, 2, 3, and 4. So this is a quick remember table. So the level of risk you see goes upwards from 1 to 4. BSL 1 is low, BSL 2 risk is moderate, BSL 3 is high, BSL 4 is maximum. So the example of pathogens that can be used here or agents that can be used here are in BSL-1 we have non-pathogenic E. coli, in BSL-2 we have Staph aureus, Hep B, inactivated HIV, etc. And in BSL-3 we have disease-causing pathogens which do have a treatment or a vaccine, and they are still can cause serious diseases like Mycobacterium tuberculosis, SARS-CoV-2, etc. And BSL-4 is very rare and it is only at particular places, like they have agents which do not have vaccines or treatment like Ebola, Marburg, etc. Then lab practices for BSL-1 are standard, BSL-2 are restricted access, biosafety cabinets for aerosols, etc. BSL-3 are controlled access, decontamination of waste must be done. BSL-4, maximum containment, full decontamination must be done. And the PPE kits that are used for BSL-1 are basic lab coats, gloves are optional. BSL-2, uh, lab coat, gloves, and eye protection is a must. BSL-3, biosafety cabinets are a must. HEPA filters are a must. Respirators are a must. BSL-4, positive pressure suits are a must. And all the other biosafety cabinets are required. And isolated facility, airlock system, HEPA filtration, chemical showers upon entry and exit. So the key points to remember is BSL increases with pathogenicity, mode of transmission, and severity of the disease. Engineering controls here are biosafety cabinets, ventilation, and airlocks are critical for BSL-3 and BSL-4. Training and PPE are essential at all levels, increasing with the risk. Waste management and decontamination are stricter at higher BSL levels. Entry and access control is minimum only for BSL-1 but extremely strict for BSL-4, and it is also done for BSL-2 and 3. Only trained personnel and limited access will be given from BSL 2 onwards. So this is how we work in different labs in BSL levels. So in BSL 1, you can see here we can work on an open lab bench or a table with uh, a lab coat and goggles and gloves. So if it is needed, if there can be any sort of spillage which can be effective. And in BSL2, all the procedures that can cause infection from aerosols or splashes are performed within the biological safety cabinet. Lab coats and gloves are worn. Eye protection and face shield can be worn as needed. In BSL-3, all work microbes must be performed within an appropriate biosafety cabinet. This is the biosafety cabinet, and the laboratory coats, gowns, eye protection, face shield, shoe must be worn, and respirators must be required. You can see they are wearing a respirator, so this they do not inhale any sort of pathogens from here. And basal for it, since it is highest, it has a separate building or is isolated, and it's present in a restricted zone, and full body air supplied positive pressure suits are worn here, so that in no way there is a leak of air, and there might be any sort of infection that can be taken up during exposure. So here you can see biosafety level. We have a simple microscope. Here we have a cabinet in bio level 2. In biosafety level 3, we have cabinet along with mask, head cap, everything, respirators. And biosafety level 3, you have a full-blown positive pressure air suits, and it's very hazardous. So this was it about the biosafety levels, and the session is done. So we can close it. That is it. Thank you.